A new luminescent sensor can detect terbium, a valuable rare earth element from complex environmental samples like acid mine waste. The sensor, developed by researchers at Penn State, takes advantage of a protein that very specifically binds to rare earth elements and could be harnessed to help develop a domestic supply of these metals, which are used in technologies such as smartphones, electric car batteries, and energy efficient lighting. A paper describing the sensor appears in the Journal of the American Chemical Society. Terbium, one of the rarest of the rare earth elements, produces the green color in cell phone displays and is also used in high efficiency lighting and solid state devices. However, there are a variety of chemical, environmental, and political challenges to obtaining terbium and other rare earth elements from the environment. Developing new sources of these metals also requires robust detection methods, which possesses another challenge. For example, the gold standard method of detecting rare earth elements in a sample, a type of mass spectrometry called ICP-MS, is expensive and not portable. Portable methods, however, are not as sensitive and do not perform well in complex environmental samples where acidic conditions and other metals can interfere with detection. The researcher says that there is not currently a domestic supply chain of rare earth elements like terbium, but they are actually quite abundant in non-traditional sources in the US, including coal byproducts, acid mine drainage, and electronic waste. In this study, they developed a luminescence-based sensor that can be used to detect and even quantify low concentrations of terbium in complex acidic samples. The new sensor relies on lamotrigine, a protein that the researchers previously discovered that is almost 1 billion times better at binding to rare earth elements than to other metals. The protein's selectivity to bind rare earth elements is ideal for a sensor, as it is most likely to bind to rare earths instead of other metals that are common in environmental samples. To optimize lamotrigine as a sensor for terbium specifically, the researchers altered the protein by adding the amino acid tryptophan to the protein. Tryptophan is what is called a sensitizer for terbium, which means that light absorbed by tryptophan can be passed to the terbium, which the terbium then emits at a different wavelength. The green color of this emission is actually one of the main reasons terbium is used in technologies like smartphone displays. The researchers developed many variants of the tryptophan lamotrigine sensor, optimizing the location of the tryptophan so that it does not interfere with lamotrigine's ability to bind to rare earth elements. These variants provided important insights into the key features of the protein that enable it to bind rare earths with such high selectivity. Then they tested the most promising variant to determine the lowest concentrations of terbium the sensor could detect in idealized conditions, with no other metals to interfere. Even under highly acidic conditions like that found in acid mine drainage, the sensor could detect environmentally relevant levels of terbium. Next, the researchers tested the sensor in actual samples from an acid mine drainage treatment facility in Pennsylvania, an acidic sample with many other metals present and very low levels of terbium, 3 parts per billion. The sensor determined a concentration of terbium in the samples that was comparable what they detected with the gold standard method, suggesting that the new sensor is a viable way to detect low concentrations of terbium in complex environmental samples.